I would like to give a brief talk about my research, and I'm mostly focusing on the cytotoxic part of snake venoms. So the toxins that destroy your tissues, your flesh, your blood vessels, and your skins. You name it. This is really the focus of my PhD. I will really shortly give a quick introduction about the problem of snake bite, and especially focusing on the morbidity of snake bite. Then I will give a short recap of traditional cell culturing assays that I've been using in the past. Then we are going to some really cool SEM data, scanning electron microscopy data. We saw already some with Colin's presentation, but this is a different way of using the SEM. And then we move from 2D to 3D models in which we use organ on a chip technology to really go into detail on the 3D structure of cells and the effects on the cells that are caused by the venoms. So really shortly about myself. Indeed, my name is Mátyás. I'm a PhD candidate, and I was fascinated by snakes all my life. On the left, we see a photo of me photographing I think it was a road-killed grass snake when I was about 7 years old. And the photo on the right is a photo that I made last year in Arizona when I visited the Venom Week conference, which was just truly amazing to see so many like-minded people that were fascinated about snake venoms and other types of venoms, and to have them all together. And we did a lot of road cruising, and here I caught a Mojave rattlesnake, and just it's amazing to see. So over almost 20 years of more than 20 years of snake experience. So this is also the topic and again of my PhD. And what I've been doing most of my time is developing a platform to study the cytotoxic effects of snake venoms, crude snake venoms, but also fractionated snake venoms. And this is important because most of you will probably know about the problem of snake bite, causing a lot of fatalities, and also more than 450,000 named victims. So people that lose their hands or their limbs, or they get blinded, or they get severe skin necrosis, as you can see in the pictures on the right. This is actually a vitis aerians, if I'm not mistaken. There is some research going on, of course, on the cytotoxic side of things, but not as much as the research is focused on more the hemotoxic and the neurotoxic side of snake venoms. So this is why this is still a very interesting topic to dive into. And this is also why I decided to focus my PhD on this. We used a selection of medically relevant species from all the regions that are most heavily affected. So these include the botrops, but also the mambas, some cobras, cytotoxic cobras, also non-cytotoxic cobras. We have saw-skilled vipers, puff adders, and some Asian vipers as well. And we developed bioassays in which we look at the live cell count, which I will elaborate on a little bit further on in the presentation. We looked at metabolism and viability of the cells, and we looked at cell morphology. So when I give a short introduction about the traditional cell culturing assays that we used a Arpitec cell line, which is a often used kidney cell line, which is very easy to grow, relatively cheap to grow, and it is really nice because it forms this amazing monolayer on the bottom of your well plate. And with these cells, we can do very easily cell viability measurements. So we use two different dyes. We have host, which is a cell permeable dye, which enters the cell when the cell is alive or is dead. So it's cell permeable, so it can enter the cell at any point of the cell being alive or dead. Then we have a cell impermeable dye, which is propydium iodide, abbreviated as PI, which cannot enter the cell only if the cell membrane is disrupted in a certain way. So what you see on the pictures here is all the blue dots are the cell nuclei, because the host stains the DNA and thereby also the nucleus. So you will see a lot of blue cells because these are all the cells that are present in your well plate. Then we have some dead cells, so it's cells that are stained, of which the nucleus is stained. But this is normal. You always have a few dead cells in your petri dish. But then if we add a venom that affects the membrane in a certain way, then this PI can more easily go in. And what you then will see is that a lot of the cells get stained. So they basically die. They go in necrosis or apoptosis, mostly necrosis, because it's a very quick effect. And what you will see over there is then this really nicely stained. It's almost like an oil patch that is all your dead cells. And this is after introduction of Naya Mozambica, so the Mozambique spitting cobra venom. So this is basically the most easy and straightforward way of quantifying whether your cells are dead or alive. This is also not only qualitative but also quantitative. Then if you introduce a venom that affects the cell membrane, you will see here, this is a zoomed out picture of your cell. So if you look very carefully, you can see hundreds, maybe even thousands of nuclei, so a lot of cells. And within a few minutes already, the venom starts doing its work, and you will see the number of dead cells increase very, very rapidly. It's even so extreme that when I would enter a concentration of 100 micrograms of the snake venoms, I would have to run to the fluorescent microscope because the effects would already be present after less than a minute. So it was literally adding the venom, then run to the fluorescent microscope, press start, and then it would take subsequent photos after 0 minutes, 3 minutes, 6 minutes, etc. And you can see already after 15 minutes, most of your cells are dead. Then when you zoom in, you can really nicely see this oil patch, as I was mentioning before. You will see it increasing over time and getting bigger and bigger. So this is what happens with your elapid venom, so your cytotoxic elapid venoms, which affect most likely the cell membrane. Then with your viper venom, this is Aegisthus salatus, which is a saw-scaled viper, we see a completely different pathology. So first of all, we see that the effect is a lot less sudden as seen with the spitting cobra venom. What you also see is that no cells are dying, the cells are doing fine apparently, at least the membranes are doing fine. But what you see is from the top left corner, you see your monolayer of cells starting to detach from the bottom of your well. And what you then see over time is that your monolayer is basically subtracting, and then at one point it moves out of focus and you will not see the monolayer anymore. So basically what we think is happening here is that the venoms, or the toxins in the venoms, that they affect the ECM substrate. So the substrate basically which glue the cells to the bottom of the well, the monolayer to the bottom of the well. And if you then zoom in a bit, you can really nicely see that this monolayer is rolling over. It's almost like your sleeve is being pulled up. Is what you see in your petri dish, which is truly fascinating, but also scary to see, in my opinion. What you can also do with these microscopes is that you can make really nice time series videos. So you take a lot of photos right after each other, and then you make a video of it. And this is what you see for the viper venom. You can see that the cells are doing relatively fine. You have a few dead cells, but then from the top left corner, you will see this monolayer moving up and then moving out of focus. So this is what happens over the time course of about 2.5 hours to your cells. And then with the Naya Mozambica, so the spitting cobra venom, we see a completely different way of the cells being affected. So you see really nicely this monolayer of cells, which is dying basically, and you see this increasing as an oil patch almost. So when you then have this still image, you can really nicely see the differences that you observe. So with the viper venom, you can really nicely see here this monolayer of cells. Cells are basically doing fine. It's even so that if you would again resuspend these cells, so you would bring them in single cell suspension and you would plate them out again, they will just happily grow further. So the cells themselves are not affected by the viper venoms, but the monolayer is. Whereas with the spitting cobra venom, there's no way that you will get these cells to happily grow again because they're basically dead. So it's necrotic and they die within minutes. Then we had some really nice SEM images of the pathology that we saw with the saw-scaled viper venom. This is basically the bottom of your your Petri dish. It's not a normal Petri dish, it's a really tiny Petri dish, 96 well Petri dish basically. And then you can already see these really nice lesions where the cells are detaching from the bottom of the well. And if you then zoom in, you can really nicely see over here that this is the bottom of your well, which is a beautiful, clear bottom. And then over here you can see this rolling over of the monolayer, and here the cells are doing perfectly fine. So you have 2 or 3 lesions over here, and then these cells are doing perfectly fine. You can really nicely see this monolayer. And then when you zoom in further, you can see over here that these are the cells that are doing perfectly fine. And this is the part where the monolayer is really subtracting. And here you have some cells that are basically they're dead, yeah, because they normally are in this nice monolayer. You can see them over here. This is a cell, this is a cell, this is a cell, and this is basically the nucleus that is detaching. And if you zoom in even further, you can really nicely see that these are probably communicational fibers, which you observe quite often with these architects, with these kidney cells. And then we move from 2D to 3D in a collaboration with Mimetas, which is an organ-on-a-chip company. What they have is a really nice, they have individually addressable chips, they call them. And what they are basically is this is a 384 well plate, and within 9 wells of a plate, there's 1 chip. And these are basically 3 tubes, which are in the middle in contact. In the middle tube, you can put your ECM matrix, and this can be any substrate. It can be a mixture of collagen, gelatin, actin, you name it, or it can be a single substrate, so maybe only the collagen. Then in the top row, you can, or in the top tube, you can put your cells. And then the nice thing is that the cells grow in a 3D fashion. So they do not grow only in 2D or the bottom of the well, but they grow entirely over the entirety of the tube, basically. So you get a 3D model of your cell growth. And the nice thing is that these plates are put on a moving table, basically. So they can even mimic the shear stress that is happening with, for example, these are endothelial cells, and then you even have a fluid that flows through the endothelial cells. So it's almost like you're looking at a near in vivo model of a blood vessel, basically. And this is what you see over here. So this is your blood vessel, and here is your ECM. And what you can do is add here growth factors, and then your endothelial cells will grow out. So this is basically angiogenesis that you see over here, and these are the tiniest capillaries that are being formed over here. So this is really a beautiful model in which you can look at cytotoxic activities of the venoms. Then some images that we got when comparing the star-scaled viper with the spitting cobra venom, but also with a black mamba. So this is your control, and you can really nicely see we have 3 different stains used here. We have the host, which is staining the nucleus of the cells. Then in green we have the actin filaments, and in red we have CD31, which is a tight junction marker, which shows where the cells are in contact with each other. So with the control, you can see your beautiful fibers, and you have also these really nice actin components that are keeping the cells together. Then with dendroaspis venom, with the black mamba venom, you see not much happening. You still see your actin filaments, you see your CD31. But then with Mozambique, you see something interesting, because you don't see the CD31 present anymore, and also something happened with the actin filaments, and we do not really know what is happening right now. But this definitely has something to do with the fact probably that the membranes are being disrupted in a way. And then with the star-scaled viper venom, we see again this really nice detachment of the cells. You get blabbing over here, which is basically caused by the actin filaments, which are being cleaved, and then the actin filaments they combine all in at one point of your cell, so they're surrounding your cells basically. And it's really nice to see that you see an overlap in the pathology that we saw in the 2D models, but also in the SEM models, and also in the 3D models. So this is really nice to observe. And we also did some therapeutic screening. So here we have a normal light microscopy image of one of your tubes. So this is again the tube. Here you have your ECM layer, and this is your tube. You're basically looking at the top of the tube. So here you have your ECM, sorry, your SARS-killed viper venom. This is the same for this one with a calcium EM stain, which is trapped within the cells. And only when the cells disrupt, this stain can leak out and it will decrease in intensity. So what you see over here with SARS-killed viper venom is that the calcium EM is still trapped within the cells, but the cells are being detached. And this is why you see this clumping of the calcium EM. Then with your control, of course, you also see no leakage of the calcium. But then with pinomastat, which is an SVMP inhibitor, you can see that you can basically rescue the cells from getting detached from each other and from their surroundings. So you can see that even with nanomolar concentrations of pinomastat, you can have the cells being in near to perfect conditions. So this is also nice that you can also use these 3D models to do some therapeutic screening. And again, this is just a preliminary data. So we're testing out more and more different compounds. Let's see. So yeah, so conclusion of future perspectives. So we successfully developed some cell-based assays that can be used to study the cytotoxic effect of snake venoms that are either crude or fractionated. I did not show any fractionated data, but this is possible. We see matching effects in the flow microscopy, also the SCM data and the 3D data, so the mimetos data. And we saw that the assays are suitable for neutralization studies, which is, of course, highly valuable. Then some future perspectives, we will be studying the effects on other cell types as well, so also including keratinocytes, so your skin cells, and hepatocytes, so liver cells, and maybe even cardiac muscle cells. And we are currently working on the development of high throughput screening assays to, in more detail, study the mechanisms of cytotoxicity. So we're looking at membrane disruption, we're looking at ECM degradation, to better visualize the effects that we observe in vitro, basically. Yep. So that is what I had to say to you guys. Thank you all for listening, and I'm happy to answer any of your questions.
Thank you very much. Super interesting and super awesome slides. And there's the first question from Ferenc.
Hi, yes. Can you hear me?
Yep, yes. Loud and clear.
Yes. So my question is that did you record on room temperature?
For the cells, you mean?
Yes.
No, it was a 37. So we have a really, it's a pretty cool fluorescent microscope. It has an incubator built in. So we have the CO2 and the temperature options available. So we don't think that the temperature really affects the result, but we haven't tested it on room temperature. We did everything at 37.
Yes, thanks.
No worries.
And I have a question. You said that you used one of the upcoming metalloproteinase inhibitors. Did you use also some more physiological ones, like the tripeptides that are known to be inhibitory for metalloproteinases?
No, we have so far just tested the sort of the more general inhibitors. Okay. So myomastat, vatimastat, and prinomastat. So all the stats basically, but we haven't tested any other compounds. If you have suggestions, please let me know, because maybe they're even more effective.
Okay, then I think we will have some further talk per email. So
that's good. That's good.
And another question is, you said that it's actin related. So the metalloproteinase alone destroys the actin, or did you test with some more fractionated venoms to see if maybe the disintegrins at earlier fractions, or maybe hyaluronidase make some effect on this rolling over? Yeah,
so that is a really good question, and also really, to be honest, a little bit of a frustrating part of the story, which I didn't show, of course, because I'm only showing the amazing effect. No, so what we see is as soon as we fractionate our venoms, We use an LC separation method, so normal C18, the first phase column, and we use acetonitrile with TFA as our mobile phase. And as soon as we add a little bit, Eric was a proud contributor to these findings, is that as soon as we add, I think it was more than 20 or 30% acetonitrile, we would lose complete activity of the metalloproteases. So with the crude venom, you would see perfect detachment of the cells. As soon as you would fractionate the venoms, you would lose all the activity for the fiber venoms, not for the elapid venoms, because we think that it is to do with the 3-finger toxin and PLA2s, which are a lot harsher. But we did do sec fractionation, and then we would still have this ECM degrading activity. So it has something to do with the mobile phases that we're using. As soon as we go for a salt buffer, we see a perfect effect. So this is still something that we have to work out in which we want to develop a way in which we can still use our old worthy C18 column. But there's no option at the moment because we're still looking into the way of solving this issue.
Did you know which kind or which subfamilies of metalloproteinases are in the Echis venom? Because maybe you have some complexes with C-type lactans or something like that. I know that the C-type lactans don't like the high pressure of the HPSCs. So there are some purification steps for the C-type lactans where you can isolate them still intact. So maybe they have some effect, but they also just did by size exclusion.
Okay, yeah, I see. Yeah, so with the SEC, it wasn't possible to really pinpoint which subtypes of SPMPs we would see. But it's actually an interesting story because a colleague of mine is now working at the same assay, but also doing proteomics. So we hopefully in the near future will know which toxins are involved and maybe also which subgroups. But I don't have any suggestions for that. It could also be a combination, of course, that we have the P1s and also the P3s. I don't know. It could be a combination of everything.